Hi everyone, I just wanted to talk a little bit about genetics with you tonight. Um, I am going to go over the Mendelian squares here for a little bit and talk about how heterozygous dominant traits can get expressed and how recessive traits and sex-linked traits can be expressed. So, all right. So here we have an assumption that this person here, we're going to say is the mother, and this person right here is the father. And we're going to look at heterozygous with homozygous and the odds of passing the trait down to their descendants. Now, our study guide has a question on it that says, what is the percentage of children, or what's the percentage of passing down a heterozygous trait? That's assuming you have one heterozygous parent and the other parent does not have the trait and that they are recessive. So what we would do is this parent would give this A to this child, and this parent would give this A to this child. So we would have a dominant trait and a recessive trait. And because this trait or disease or whatever it is dominant, this child will express the disease. And so, once again, if we were to go over here, if they gave this chromosome, since they're heterozygous, and this parent is still giving this one, we are not going to express the trait. We're not going to have it because they only got the regular chromosomes. And then here, this dominant one would go to this child, this recessive one would go to that child. And so we would have two children that would express the trait. And then last but not least, we'd have two non-dominant traits going for this last square here. And so this kind of breaks it down and it gives you an idea of what the percentage of, if one parent has a heterozygous dominant trait, what the chances that their offspring would have that trait as well. So it would be 50% because it would be 2 out of the 4 boxes, which would be 50%. So moving on, if we were doing autosomal dominant traits still, and now we had 2 heterozygous parents, like I said, the study guide is assuming that only one parent has the trait. You're going to have some changes and it's going to increase. So these big A's will come over here and this child will have the trait. These big A will come over here, small a will come over there. They will express the trait as well. Same here, big A comes down here, little a comes over here. They express the trait. And then their last child would not express the trait. So it's a 75% chance if both parents are heterozygous autosomal dominant that their children would express the trait. And so on, we would have the same thing. If the parent was homozygous and another one was heterozygous, you would see all of the children would express the trait. And then once again, of course, if both parents are homozygous autosomal dominant, they would all be homozygous autosomal dominant. So now let's look at those recessive traits. Those recessive traits would require 2 of those chromosomes in order to express that trait. So this could be blue eyes, this could be some kind of disease, it could be something like that. So we're starting off with one parent that's heterozygous, so they have one dominant, one recessive trait. And we're assuming this one is a disease trait or something that we're interested in mapping out. So, and this parent doesn't have the disease trait. This one would be called a carrier because the big A would be the dominant trait and it would supersede the recessive trait. So they would be what's called a carrier. This person would not have the trait. They would not express the trait though. So this first square, we would have two big A's. So they are homozygous and they do not have any recessive trait. So this is only dominant. So they would not express whatever trait it was, whether it be blue eyes or some kind of disease. Now the next square, we're going to get an A from here and an A from here. So they're going to be heterozygous as well, just like this parent right here. Now this one is still dominant, so it's not expressing the trait. They become carriers. And then we're going to get a big A and a big A here, because this one is not a carrier. So two big A's, they don't have the trait. And once again, big A and a little a. So in this case, half the children will not be carriers and will not express the trait. The other half will be carriers but will not express the trait. So here we have two parents that are heterozygous autosomal recessive. So these parents are called carriers. Both of them are carriers. So we're going to take the big A's here. We're going to take one big A, one little a here. So this one becomes a carrier. This is not a carrier, and it does not have the trait. This is a carrier. It does not express the trait though. Once again, big A here, little a here. So this is also a carrier. And in this last one, two recessive chromosomes or genes are coming together, and this one is it's not just going to be a carrier, but it's going to express that disease or trait that we are looking at. So in order for this to happen, you must at the very least have parents, both parents must be carriers. So that is a review question as well. So here's another example. If one person had the trait, one parent, and one person was a carrier, then they would also pass along this trait or disease. But the percentage is going to change a little bit. So we get the big A from here, little a from here. Now we have two little a's, big A and a little a, and two little a's. So half of their kids are going to express that trait and half will be carriers. So now we're going to move on to sex-linked. Now this is normally how a healthy sex-linked chromosome would pair up. So you get, this is dad because there's a Y chromosome up here, and here this is mom because there's two X's. So we could pair up these X's. We could pair up this X and that Y, and this would be a male. This would be a female, of course. These X's would be a female. X and Y would be male again. All right, so this is how it normally should occur. Now we're going to look at some abnormalities. So let's say the father has a disease, and it's X-linked. The disease is located on the X chromosome. So he's expressing the trait. He's not just a carrier because he doesn't have a backup X. Now, if he were to have children, this could be a possibility. We'd have one healthy X from mother, one trait or diseased X from the father, and that would result in a female offspring, but she has a backup X. She may be a carrier, but may not express that trait. Now, if the father gave the Y, that means only the mother can give the X at this point. So this child, who's a male, would be fine because the male can only get a Y from their father. Now here the father is giving this X, the mother is giving this X. So once again, this daughter is a carrier, but does not, most likely will not express the trait because she's got a backup X here. This last one, once again, that son will be fine. Now let's look and see what happens if it was the mother. So here This time the mother has an affected chromosome, one of her X chromosomes. We're going to look at the percentage of what would happen here. So these X's would come together. Once again, we have a backup X, so this daughter would become a carrier as well. This is the only X, though, given to the son, so the son would then express the trait. This is why males often express sex-linked traits and females do not, because the females have a backup X. Here, this daughter is fine. She does not have the trait, and she is not a carrier. And here, this son is fine. He does not express the trait. So now we're going to talk about non-disjunction of chromosomes, and we're going to look at the sex chromosomes in particular because a lot of, when it comes to monosomy and autosomal chromosomes, the offspring will not survive in most cases. Now, you could have extra chromosomes like trisomy 21, which is Down syndrome. But we're going to look at the sex chromosomes because I think that's a little bit easier to understand sometimes. So we're going to start off with our whole process of meiosis. Meiosis 1, meiosis 2, and that is a reductive process, if you remember. So we should have 23 pairs of chromosomes. So we would have 46 in all. And through meiosis 2, these would be reduced. This would divide, and we would have this X go over here, and this X go over here, this chromosome. These are pictures of chromosomes here. So it would separate into two separate gametes. Okay, remember meiosis only occurs for those gametes. It creates either an ova or sperm cell. So here, once again, we have another example. So this is how it's supposed to happen. So we're going through meiosis 2 again, and we're dividing. And imagine like there's 23 chromosomes, but we're only 23 pairs of chromosomes, but we're only looking at the X and Y right now, and this would be a female. So it's going to divide. But what ends up happening is the cells dividing, these don't separate for some reason. It's called nondisjunction. It happens more often in females in their ova than males, so it's often a female problem here. So we have nondisjunction, but we still form two separate cells. So the rest of the chromosomes are going to be in here. So we'll have like 22 instead of the 23. So this was supposed to split right here, and it didn't. So we're going to look up at this in more detail. So let's say I have an ova here, and it has an extra X chromosome. This is going to be missing information, genetic material. And since we're looking at the sex chromosomes at this moment, we are missing an X chromosome. Okay? So let's look at this first chromosome that has an extra one. Okay? So we have this. This is going to be, let's say, our ova. Okay? And over here, we have another individual. This is going to be dad. And dad is going to go through meiosis 1, meiosis 2, and it's going to be reductive, and it gets split. Okay? So dad has an X and a Y in the sperm cells, and our zygotes, and they're haploid cells. They only have 23 chromosomes, not 46. There are gametes. And now fertilization is going to happen. So we're having a boy. So we have the X and the Y are coming together, and they move along, and they join, and conception just happened, and it got fertilized. But notice that this is going to be X, X, Y as a result. So this makes it non-disjunction happened, which led to this XXY, and we end up having Klinefelters. So we have an extra chromosome. So we're supposed to have 46 chromosomes, right? We have an extra. We have 47 now, okay? And so it's going to be coded, Klinefelter's is going to be 47 and then XXY, because there's two X's, all right? Because it's with the sex chromosome, so we're going to acknowledge that. Now let's look back. So we said that this was supposed to separate, and we were supposed to have one X here and one X here. So whatever happened to this ova that formed? If there was an ova that formed, whatever happened to that? We're going to look into that and try and figure out what's going on. So here we're gonna pretend Dad, or that could have been, who knows what that was, but Dad gave an X this time. All right, and they start to join. So here's the sperm, here's the egg. Oops, and slide closed here. So the sperm and the egg join, and you notice we only have one X chromosome now. We don't have a second chromosome. Now, because the Y doesn't have a lot of genetic material on it, we're fine. I mean, not fine. You're still missing some genetic material, but the person's going to be able to survive. So this is going to be Turner syndrome. And it's going to be presented, there's short one chromosome, so instead of 46, they have 45. And it's going to be labeled XO because they're missing that other chromosome. So I hope this cleared up a little bit about the differences in chromosomes and how autosomal dominant works, recessive, sex-linked, and nondisjunction. So good luck.